Okay, happy Tuesday, grade 12s. I've asked you to complete questions 1, 2, 4, and 5 on page 345, so I'm going to take that up so that you have your correct answers, okay? So it says define the following terms. A mutation is a change in the genetic sequence. A frame shift mutation is when we add or subtract a nitrogenous base, so the whole frame is changed. Okay, so in other words, we're adding a nitrogenous base, 1, 2, 3 maybe, or we're deleting those. A point mutation means that one nitrogenous base is substituted for another. So in other words, we're putting the wrong nitrogenous base in. A nonsense mutation changes a nitrogenous base to make that codon read for a stop codon. Okay, so we put a stop codon in prematurely. A missense mutation, okay, means that we've changed the nitrogenous base and we're not reading it properly anymore. So in other words, a missense mutation causes a change in the amino acid sequence. Okay? And then question 2 says, the normal form of a gene contains the nucleotide sequence, and they give us the sequence. And then they ask us, what type of mutation would each of these 3 things be? So 3 extra adenine nitrogenous base pairs are inserted in positions 10, 11, and 12. So if you just go at 10, 11, and 12, they're going to add 3 adenines. So I would say definitely based on your grade 12 information that would be a frame shift and a missense mutation, okay, based on an insertion, because you're inserting things. But don't worry too much about the insertion. Just the question asks you what is going to happen. It's going to give you a frame shift mutation, which will likely result in a missense mutation. It will definitely result in a missense mutation. Okay, the third nucleotide is changed from an adenine to a thymine. So if we go 1, 2, 3, that's an easy one to find. That is changing from adenine to thymine. And what that's going to do is when we read this and we make the mRNA, that's going to give you a stop codon. And so that's going to be a nonsense mutation, okay? The thymine molecule in the 10th position is removed. So if we remove anything, if we get rid of any nitrogenous base at all, then that's going to cause us to have a frame shift mutation, okay? So question 4. Which of the following amino acid changes can result from a single base pair substitution? Explain your reasoning. So for this, you need to have your genetic code handy, all right? So if we're going to go from, sorry guys, just hit the phone there. If we're going to go from Phi to Lu, all right, the question's asking, can it result from a single base pair substitution? So yes, it can, because Phi and Lu are right beside each other. And we have UUU, UUC for Phi, and UUA, UUG for Lu. So if one of these 2 changes, if C changes to A, or C changes to G, or G changes to C, or G changes to U, etc., then that's going to cause a change in amino acids, and it only requires 1 change, because U, the first nitrogenous base, and the second nitrogenous base of the codon are the same for both of these. So that's a yes on that one. B, isoleucine to THR. So can we do that? So isoleucine is right here. Okay, just think I have to pull this up a little bit. All right, isoleucine is right here. THR is right there, thyroxine. And so we're going to have AU, AC, but we've got 3 of the 4 possible base pairs in the last codon, and we've got 4 of them here. Is it possible for one base pair to change isoleucine to THR? It technically is, because if you had AUC and you changed it to ACC, that would be just 1 base pair changing. I realize that you've got the G in this position that gives you met, but technically the answer is yes. It could require just 1 base pair, and that would be one of these 2 base pairs to change, and you could go from this to this or this to this, okay? So it is possible. All right, and that was question B. Question C, serine to arginine. So serine is over here, arg is all the way over here, And so because we start with a U and we start with an A and then we have a C next and a G next, the answer is no. That one cannot take one base pair change and become a different amino acid, one of those two different amino acids. And ASP to glycine, okay, just looking for ASP right here. All right, so we've got the GAC or GAU as our answers there. And to glycine, all right, glycine, sorry guys, G, G, and all possibilities. Okay, so technically, yes, this could happen. ASP could turn to glycine because you could have GAC, and if you substituted GGC instead of GAC, you would have glycine. So it is a possibility for that to happen. So that's a yes on that one as well. All right, and lastly, question 5. During the translation of a molecule of mRNA, the process is stopped prematurely. What type of mutation has occurred? What type of codon has been read by the ribosome? So what type of mutation has occurred? It's definitely going to be a nonsense mutation because we formed a stop codon. All right, I hope that helps you guys out.